Okay, so over 100 years ago, there was a German neuroanatomist named Corbinian Brodmann who examined human cortex under the microscope, and he, looked at how there were areas that were sort of consistent in how they looked, actually, in what he called their cyto or cellular architecture, and then all of a sudden there'd be some kind of a boundary, and then beyond that, the next area of cortex looked different in terms of its cytoarchitecture. And just to give you an example, if you look in at the most caudal aspect of the cerebrum here, right? This here's the longitudinal fissure, right? And if we were to go in, between the two occipital lobes here, along the occipital lobe on each side, medially, you'll find a sulcus. It's kind of a deep sulcus. It's sometimes referred to as a fissure too. It's called the calcerine sulcus or the calcerine fissure. And the cortex there is actually where all of the projections from the thalamus that are carrying input from the eyes, is going to arrive in synapse. So it's called primary visual cortex. And primary visual cortex, or V1, as it's also referred to, if you do a cross-section, we're going to see that it's neocortex, like a lot of the stuff we're seeing on the side here, and etc. But it's got 6 layers, it turns out. But the 4th layer down in the gray matter, like if you go in through, a gyrus, a V1, you'll hit layer 1, layer 2, layer 3, layer 4, layer 5, layer 6, and then you'll hit the white matter, basically underneath that. But the 4th layer down, layer 4, in primary visual cortex, is extremely thick. It's very thick because we're going to see that layer 4 in neocortex is the primary sort of input layer, for synapses, that are arriving, from other locations. So there's all this white matter underneath the cortex at the surface, right? And this is going to be like, for example, axons coming in and then synapsing with neurons that are located in the gray matter of V1 at the 4th layer down. The layer 4, we're going to see, again, it's going to be the input layer. So there's so much coming in through our eyes. We're exceptionally visually oriented primates, actually, that there's this tremendous amount of thalamic, projections, arriving in layer 4 at primary visual cortex in the, calcerine fissure back here, in the occipital lobe, that it looks thick. And it looks actually there's a famous Italian neuroanatomist who noted this, what he called it became known as the line of Gennari. And when you got to the edge of primary visual cortex, that thick line of Gennari drops off. So that's a pretty clear cytoarchitectural difference that actually reflects a functional, organization of the brain. The fact that we're very visually oriented, and there's a lot of visual input, sort of arriving, at V1. Now, what's interesting is that Brodmann identified 52 distinct regions based on cytoarchitectural differences, and he numbered them. So, primary visual cortex, back here in the caudal region of the brain, and medially in here, along that calcerine fissure or sulcus, is referred to as Area 17. And Area 17, primary visual cortex, lots of thalamic input, that originated initially in your eyes, arriving in layer 4, giving that very, very distinctive thick line, at the 4th layer down. But there's 52 regions total that he identified. And more recently, the Allen Institute for Brain Science up in Seattle and other institutions have been examining, the fine, microstructure, the functional architecture using various imaging techniques, for example, differences in genetic expression, using finer tools basically to further subdivide, a human cortex into, more subdivisions. So Brodmann, in some ways, identified kind of like the state lines or the national boundaries, and the Allen Institute and other organizations using more modern and refined techniques are now finding additional structural and functional kind of divisions within those specific Brodmann areas that were identified over 100 years ago. So one of the things I like to emphasize about Brodmann and about these efforts by the Allen Institute and others is that there's a tremendous amount of cytoarchitectural diversity, structural diversity, functional diversity, in the cortex of the brain, including within the neocortex of the brain, which again is the newer, in an evolutionary sense, that makes up 90% of the cortex that you see here, that we'll find here. However, even with all that tremendous diversity, in terms of cytoarchitecture, there are going to be some consistencies too across, most regions of neocortex. For example, in the layering, there's always going to be 6 layers. And also we're going to see in the patterns of inputs, like for example, in primary visual cortex, the thalamic projections, the incoming axons that are going to synapse in, primary visual cortex in Brodmann Area 17 are actually going to make their initial synaptic context largely in layer 4, the 4th layer down. And we're going to see that 4th layer down is pretty much the input layer, the primary sort of input layer for all these other regions of neocortex as well. We're going to see there's also some consistency in the outputs. The axons that we're going to see are going to be leaving specific areas of neocortex to go to other places. So despite all this extraordinary diversity in terms of, cytoarchitecture that Brodmann initially identified over 100 years ago and that's being expanded, at the same time, we also have some consistency in terms of the circuitry, that defines particularly neocortex. And we're going to be talking a little bit more about that next.